This is the introduction video for Chapter 10. This chapter, we're going to talk about DNA. We've talked about DNA in terms of chromosomes. We've talked about DNA in terms of genetics. And now we're going to look at DNA as an actual molecule and how it's built and how it really functions. A lot of what we've learned about DNA comes from these little boogers. These are viruses. Viruses are a unique organism, if you will, because they're basically DNA with a protein shell, with protein wrapped around it. And we have been attacked by viruses, but then we've also taken those viruses and used them for human gain and understanding. So it's kind of a relationship, if you will, with us and viruses that's complicated because they attack us, but then we learn from them and then we use them to create something else, and then they adapt to that and then they attack us again. So DNA as a molecule is made of one of our 4 types of organic molecules, nucleic acids. And what's important about it is not the caloric value. We learned about in cellular respiration how carbohydrates, how proteins, and how lipids have caloric value to them. They can go through cellular respiration through glycolysis, Krebs cycle, electron transport, and produce usable energy in the form of ATP. Nucleic acids have no caloric value to them. So their importance comes in their stored information, their genetic information. This is why DNA is so important, because it's holding on to the code that tells us how to build proteins throughout our body. And as we learned in the previous chapters, that's how we pass on our information from one generation to the next. DNA and RNA are both examples of nucleic acids. You may have not heard of RNA before, but you will by the end of this chapter. RNA is another form of genetic information that is used to build proteins with, or as part of that structure of the ribosomes coming together and proteins being produced. The other part of DNA, besides just the nucleic acids or the nucleotides, is the sugar phosphate backbone. So we see here kind of a picture that demonstrates the molecular components of DNA. And usually a DNA molecule looks really complicated and it has all these parts and pieces. It's really not that complicated. We have this sugar phosphate backbone right here. The phosphate is represented as a yellow, the sugar is a blue, and we see it repeats. Sugar, phosphate, sugar, phosphate, sugar, phosphate. And in terms of DNA, we have 2 strands of that. And then in between, we have the nitrogenous base. And this is the nucleic acid component of this. And that's really all DNA is made out of. There it is up close. Here is a space-saving model that kind of shows you this. This is a single-stranded piece of DNA, but you can still see the sugar and the phosphate, the blue and the yellow alternating here as kind of the backbone. And then the greens, reds, and oranges, these are the nucleic acids or the nucleotide part of DNA. Those nucleotides are very, very important to the code of DNA. This slide right here, this information is very important. I want you to put a star here, highlight it, circle it. This is really important for you to know. There are 4 letters in the codes of DNA. These 4 letters, it's an alphabet that makes up every protein in your body, but there's only 4 letters in this alphabet. This code is not complicated. This code is very simplistic in that it only uses 4 letters. So we're going to talk about these letters a lot during this chapter. The 4 letters that we have are thiamine, which is represented with a T, cytosine, which is represented with a C, adenine, A, and guanine, G. Those 4 letters, A, T, C, and G, are all found in DNA. And as we continue to talk about RNA, we're going to learn about another nucleotide that's called uracil. represented with a U, and it is found, uracil is only found in RNA, and it replaces a thiamine that would otherwise be found in DNA. So this is very important. When we start looking at strands of nucleic information or genetic material, if you see in the code the letter U, then you know we're dealing with RNA. If you see the letter T, then you know that we're dealing with DNA. So we talked about Mendel during the last chapter and how important he was in discovering these patterns of genetics, and he's the father of Mendelian genetics. So now we're going to learn about Watson and Crick and their discovery of the double helix. So James Watson and Francis Crick are credited with determining the double helix shape of DNA. So the double helix is a very, very important part of the molecule, the shape of the molecule. People knew before Watson and Crick, people knew the basic components. They knew about the sugar and phosphate. They knew about the nitrogenous bases, A, T, and C, and G. They also knew that the amount of A and T were always the same, and the amount of C and G were always the same. What they didn't know was, how are these molecules put together? What did they look like? This picture here is an extremely famous picture. Almost any biology textbook will have this picture in it of James Watson and Francis Crick. So this is Watson, okay, and this is Crick. And what you see in this picture is a model that they have built out of organic molecule components, like you might use in an organic chemistry class. And this is DNA. So these two scientists are credited with discovering the double helix shape of DNA and finding what DNA is made out of. Not made out of, I should say, but how those pieces that it's made out of are put together. This is in my opinion, the greatest discovery of the 20th century. The reason that I say that is the application of what we're able to do from their information is unmatched. A lot of people might say that Einstein's theory of relativity, has really changed it, and it has. But it changed our world in an applicable way. What Watson and Crick came up with for the structure of DNA has changed our world all around us. And we're going to talk about that in the next chapter when we look at DNA technology. So their discovery, not of DNA, but of the shape of DNA, has completely changed our world. But it's not without drama. There's always drama. Watson and Crick needed help in understanding the shape of DNA. So they turned to a fellow scientist, Rosalind Franklin, who specialized in X-ray crystallography, which means she was able to use X-rays to look at the crystalline structure of molecules. This is before we had use of electron microscopes, and it was a way, taking these special kind of pictures, to try to understand the molecular structure. The problem is that at the time that this research was being done and X-ray crystallography was being used, we did not understand that X-rays were mutagenic or carcinogenic. So Rosalind Franklin spent hours and hours and hours taking pictures of these DNA molecules, all the while being exposed in an unprotected way to these X-rays. And actually what happened is she died. She died of cancer that was the result of her using all of these X-rays. Here's the picture. Now to you and I, it looks like some kind of, fuzzy thing on your television screen, which means you aren't getting a good signal. But to Watson and Crick, this was it. This was that last piece of information that they needed to put everything together. They saw this picture and they recognized the double helix shape. And so they were able to put what they had already come up with, what others had come up with, and put that all together and propose this double helix formation of the genetic material. This discovery was published and they were awarded a Nobel Prize. The problem is that Rosalind Franklin had passed away by the time the Nobel Prize was awarded, and the Nobel Prize cannot be awarded to somebody who has passed. So you will always see Watson and Crick's name published and credited with the development or the understanding of the DNA molecule, but you don't see Rosalind Franklin's name on the Nobel Prize. But you do see her name and her picture in almost every biology textbook. So it's important that we recognize what Rosalind Franklin did, especially as a woman scientist at that time, and the fact that she literally paid for this discovery with her life, and remember that. So here's what they were able to come up with. By looking at Rosalind's picture, Watson and Crick said that DNA is shaped like a rope ladder that is twisted. The stiles of the ladder, okay, the part that you put your hands on the side, those are the sugar and phosphates that are repeating in that pattern. The steps or the rungs of the ladder are the base pairs, the A's, the T's, the C's, and the G's. It's really not that complicated. Here is our rope ladder, and you can see the concept of twisting it to create a double helix. This is what has changed our world. And here's the other part that really brought this picture together. We knew about A, T, C, and G, and we knew that the amount of adenine in DNA always equals the amount of thymine, and the amount of cytosine always equals the amount of guanine, but we never understood why. Now what we understand is that adenine always pairs with a thymine to create one of the rungs of the ladder. It is always A and T together. Always A and T together. So in lecture, I'm going to try and say this as many times as I can. A always pairs with T. Always. We also see that cytosine pairs with guanine. That forms another kind of rung or step for our ladder. So C always pairs with G, and A always pairs with T. Always. This is extremely important. We knew that these amounts were the same in DNA. We just couldn't envision how they were put together. But the double helix helps us to understand how these molecules were paired together. This leads us to a movie that came out in the 90s called Gattaca. Maybe you've seen this movie, maybe you've heard of it, with Ethan Hawke and Uma Thurman. But this movie is all about genetic testing and how it is used to determine how is your health going to be in the future? What kind of job are you going to have? It creates a class system. People are judged based on their genetics as soon as they're born. A DNA sample is taken, they're analyzed, and they're immediately put into two different groups, a higher ranking and a lower ranking. And that's kind of what the premise of this movie is. But if you look at this picture, which is the cover of the movie poster, you notice the staircase. Notice the two characters here running the staircase. It's the double helix. Also, notice the name of the movie, Gattaca. A, T, C, and G. These are all letters that DNA is based on. If you haven't seen this movie, I highly recommend it. It's really good. Maybe even better than Twilight. All right, so here is how these pair up. So I told you A and T and C and G always pair up that way. Here's why. It has to do with the hydrogen bonds. Remember, hydrogen bonds are weak bonds. Remember I said they're like Post-it notes? They can kind of stick and unstick. This is where that's key. This is where that's really, really important. We see that the way that adenine and thymine are built, they can form two hydrogen bonds. The way that guanine and cytosine are built, they can form three hydrogen bonds. That is why it is always A with T and C with G. Always A with T and C with G. And that's it. That's DNA. It's not that complicated. Phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, A to T, C to G. That's it. It's not a complicated code, but it's a long code. Our DNA is extensive. Our chromosomes contain huge amounts of these base pairs. That's where the excitement of the code comes from. If you took our DNA and you stretched it out, okay, and you were reading the letters, the A's, the T's, the C's, and the G's, if you recited the entire 3 billion genome letters at the rate of 100 A to T, C to Gs per minute, without sleeping, eating, or drinking, it would take you 57 years to recite your DNA code. That's the awesome power of DNA. The code is simple. A to T, C to G. It's the length of that code that provides all of the power. And with that, we're going to stop and we'll continue in lecture.